Start the talk.
Okay, great.
Okay, it's here. Greetings and a very good afternoon to everyone present here. I'm Vidya Rashmi Haneli, member of Proteus, the biology club ISER TVM, and I shall be your host today. We at Proteus are very pleased to welcome you all to our next faculty talk series. Our speaker for the day is Professor Zoltan Ivics, Head of Division Medical Biotechnology at Paul Ehrlich Institute, Germany. Professor Ivics received his PhD in molecular biology. After postdoctoral studies at the University of Minnesota in the USA and the Netherlands Cancer Institute, he was appointed as a research group leader at the Max Delbrück Center for Molecular Medicine, Berlin, Germany. He was appointed as head of division at the Paul Ehrlich Institute in Germany in 2011. Dr. Ivics' major scientific achievement is the molecular reconstruction of the Sleeping Beauty Transposable. Technologies based on Sleeping Beauty gene transfer have been revolutionizing genomic manipulations in vertebrate species. He received recognition of the Molecule of the Year in 2009 for developing a hyperactive Sleeping Beauty Transposase that opened the door for clinical applications. He has been serving as a member of the European Society of Gene and Cell Therapy Board and as a faculty member of the Center for Cell and Gene Therapy program in Hessen, Germany. So kindly keep your microphones and video switched off all through the session. Now, I would like to hand over the virtual mic to Professor Ivics. Thank you.
Yeah, thank you. Am I being heard? Do you hear me?
Yes, sir.
Okay, great.
Yes, sir, we can hear you
first of all,
properly.
thank you. Yes, thank you. Thank you. So first of all, thank you very much for this invitation. I'm glad that we have this opportunity to share our knowledge and disseminate interesting new research data and innovations to remote places through technology, although I have to admit I was hoping to use my presentation mode maybe a little bit easier, but it didn't work out. Nonetheless, thank you very much again for the invitation and for the kind introduction, and I will guide you through this topic of using transposable elements for genetic engineering in animal cells and in human cells, especially one particular transposon that I will introduce to you in a minute. Let me just, first of all, give you the background on how transposable elements work in nature. What do we mean by transposition, transposons, or transposable elements, and what are the basics of this biochemical reaction that we call transposition? A transposon is a jumping element, a jumping genetic element that I highlighted here, and transposition occurs during a donor chromosome and a recipient chromosome. Transposition really is a translocation process. In other words, a genetic element relocates from one chromosome to another or to a new location on the same chromosome. How does this occur in real life? The way it occurs that there are two fundamental components of any transposon, practically speaking. A protein component that I highlighted here in orange color, which is the transposase. That's an enzyme that is important for the transposition reaction. And DNA components that flank the transposon here on the left and on the right that are called terminal inverted repeats. So what happens during transposition, that this transposase enzyme is present in the nucleus. It will bind to these terminal inverted repeat sequences on the left and on the right-hand side of this transposing unit, so to speak. And after that, this protein catalyzes the actual transposition reaction, which includes removal of this gene out of the donor chromosome and permanent genetic integration of this transposing unit into a new location in the same genome. So the donor chromosome becomes empty, so to speak. So the transposon basically left this location and it transposed or hopped over to a new location on a new chromosome. That's what we call cut and paste DNA transposition, a cutting step which removes the transposon, and the paste or pasting or an integration step through which the transposon integrates and translocates to a new site. Now, one way of using this biochemical process for genetic engineering is really to incorporate a foreign genetic sequence between these terminal inverted repeats. This can be, practically speaking, anything, any kind of gene sequence, and then expressing conditionally the transposase in a cell. And then we typically maintain this kind of artificial transposon in a plasmid vector. And then out of this plasmid, this gene of interest, genetic sequence, will be excised or cut out by the transposase, and the genetic sequence will be incorporated into the genome of the cell. This is the biochemical reaction through which we use a transposable element for genetic engineering in many different types of cells. Now, one really important aspect of everything that I'm about to say is, well, first of all, the topic after this kind of introductory slide of the entire presentation will be one particular transposon, which we call Sleeping Beauty Transposon. And this is the transposon that you already kindly introduced in your introduction. And just for you to kind of appreciate what was the invention with this transposon, let me point out that this reaction that I just highlighted on the previous slide very rarely occurs out there in nature, because the transposon components, the transposase encoding gene or the transposon inverted repeat sequences, are typically mutant. They have undergone a lot of mutations. These transposons were active millions and millions of years ago back in evolution, but they are not really hopping any longer in nature. So what we really did, experimentally speaking, is that we fixed the mutation in a family of transposons. Practically speaking, we did like a sequence alignment of the transposase coding region, looked at sequence conservation, basically across the entire polypeptide of this transposase. And then we removed these inactivating mutations that I highlighted here with these yellow triangles, stop codons, frame shifts, missense amino acid mutations all over the place in this gene by mutagenesis. So basically the argument has been that we can generate a consensus sequence out of many individual genes, and that this consensus sequence would likely represent an active enzyme or an active polypeptide sequence. And this was really how we made this gene, Sleeping Beauty Transposon. And that's exactly the reason why we named it Sleeping Beauty, because we argued all of these sequences, the gene sequences, have been kind of sleeping or dormant in natural sources, in natural genomes. And we, practically speaking, used molecular biology, genetic engineering technologies to awaken this gene and to reactivate this gene and its transposition process. So really is like an ancient life form, so to speak. I just use this fairly well-known movie from Hollywood where they reconstructed entire species and organisms out of ancient DNA. Of course, a transposon is not a species, it's just a gene, a hopping gene. But nonetheless, the idea or the concept of actually reversing evolution and reawaken or reconstruct something that existed long time ago, practically speaking, the same concept or the same idea. Now, here is something really important, kind of a technology-oriented slide that shows to you how do we typically use a Sleeping Beauty transposon in a gene delivery experiment? It's a little bit like a recap of my very first slide. The idea is that we're cloning a gene of interest, here shown in the green color, between the terminal inverted repeat sequences that are typically maintained in a plasmid. So you have a plasmid, a kind of a typical cloning plasmid. The terminal inverted repeat sequences are represented there, some restriction sites, restriction enzyme cleavage sites. Between them, we clone a gene of interest between them. Now we have a new plasmid sequence, and then we supply the transposase either as recombinant protein or synthetically produced messenger RNA or as an expression plasmid. And then we take, typically speaking, two plasmids, one transposase plasmid, one transposon plasmid that has this gene of interest, and we co-transfect these two components into cells so that the transposase would be available for the transposition process. It will interact with the terminal inverted repeat sequences, would cut out this gene of interest out of the plasmid, and then would integrate this into the target DNA. And target DNA is typically the genome of the cell where we introduce these gene delivery molecules. Let me give you a little technical or methodology summary, so to speak, with respect to how the system works. Efficiency, and already the kind of introduction kind of introduced the aspect of hyperactive transposases, and that we generated some of these Sleeping Beauty transposases a couple of years back. What are hyperactive transposases? A hyperactive transposase enzyme is a mutant or a variant polypeptide that can do the transposition reaction more efficiently, so to speak. That's the idea. So we introduced a couple of mutations in the Sleeping Beauty transposase gene and then scored transposition of these variants, these mutant polypeptides in the context of gene delivery. And through this procedure, we identified one particular enzyme that is called SB100X. This is shown on the right-hand slide in comparison with a couple of other variants and the originally reconstructed or resurrected Sleeping Beauty transposase. And as you can really appreciate, this SB100X, and we really gave this kind of name to this variant, can do gene delivery under certain experimental conditions 100 times more efficient than the originally reconstructed Sleeping Beauty transposase protein. Yeah, so that really means here's a kind of a little highlight, a little visual representation of a cell culture experiment where you take a Sleeping Beauty transposase, either first generation transposase or SB100X hyperactive variant, and co-transfect this transposase together with an antibiotic resistant gene tagged transposon into human culture cells, and then you put these cells under antibiotic selection. So all of these colonies that are stained on these dishes are genetically engineered cells that contain genomically integrated transposon as a result of an active transposition process. And as you can appreciate, this hyperactive transposase can do this reaction far more efficiently than the first generation transposase. So that's about hyperactive transposases. How about the fate of that gene of interest that you want to really move into the genome? Will this be expressed? Obviously, whenever we genetically engineer cells with a gene of interest, we're also looking forward to having long-term expression of that gene of interest. This slide summarizes the data that we generated in tissue culture by really making an entire panel, almost 300 individual cell clones that contained a GFP gene under conditions that every single clone has only one transposon integration, just one copy of the gene sitting somewhere in the genome. And control experiment here is just plasmid integration, kind of random integration of plasmid sequences into the genome. And then we score silencing, basically loss of transgene expression, GFP expression, over time. And as you see, out of almost 300 clones, Sleeping Beauty Transposon engineered clones, around 2% of the cell clones have undergone silencing. In other words, loss of GFP expression over time. And as you see, over 25%, a quarter of the cell clones that were generated by straight plasmid integration have undergone really loss of gene expression. So that kind of tells you that maybe not every single Sleeping Beauty transposon integration, but certainly the vast majority of these integrants not only permanently integrate or is permanently integrated into the genome of a targeted cell, but after genomic integration, almost all of these integrants will have the capacity to express the genetic cargo in these cells for a long period of time. Now, really the third slide that I wanted to share with you is to kind of highlight how we actually introduce these components into cells. As I have already shown, we typically work with plasmids. These are kind of classical non-viral gene delivery tools. One can really just mix together two plasmids, one for the gene of interest, one for the transposase, as I already showed. One can also kind of combine two expression cassettes on the same plasmids. One can also use recombinantly produced transposase or messenger RNA for the transposase and simply mix this together with a gene of interest. In cell culture, we typically rely on various transfection reagents, or in case of primary human cells, we're using electroporation technology to really bring these vector components into the targeted cells. And finally, it is also possible to kind of package these nucleic acid components, these DNA components of the transposon, into viral gene delivery tools, for example, adeno-associated virus or adenovirus, to bring in the transposon components very efficiently into targeted cells. Now, this particular little integration machine, so to speak, very simple enzymatic reaction, but very powerful with respect to just sticking a piece of DNA into the genome, really opened up quite a number of areas in genetic engineering. For example, in cell culture, one can just use the Sleeping Beauty transposon for permanent genetic modifications of cell lines in cell culture. Germline transgenesis, what really means is the production of genetically engineered animals for biomedical research or for agriculture or for biotechnology, really. Functional genomics, what this really means is that we can use the transposon to purposefully knock out genes or overexpress genes in an organism, for example, in the mouse. And then this can lead to certain phenotypic changes that the investigator can observe and then can identify the affected genes that lead to an observed phenotype. And this has been fairly widely used, typically with the Sleeping Beauty transposon system, to drive cancer in the mouse system in different somatic tissues, with the goal of identifying new genes, proto-oncogenes and tumor suppressor genes that play a fundamental role in the tumorigenesis in various cancers. And finally, gene therapy is just the opposite, where we're using a transposon vector to really fix a certain damage or a certain mutation or a phenotypic problem in a certain genetic disease. And I will spend a fair bit of my time towards the second half of this presentation on the gene therapy applications of sleeping beauty transposition. But first, let me just share with you maybe 2 or 3 slides with respect to germline gene transfer, because this really highlights how efficiently this reaction can work in a real-life experimental system. Let me just quickly kind of highlight to you a technology that has been used for germline transgenesis in mammalian species, in animals. This has been established for the mouse, maybe late 80s, more or less. The technology is called pronuclear microinjection. What this technique really is that you fill up kind of a fine needle or a microcapillary with naked nucleic acids. Traditionally, it's plasmid DNA. And then you take a freshly fertilized mouse egg, a zygote, and physically you kind of inject your nucleic acid into the nucleus of that zygote. So this is called pronuclear microinjection. So it's very simple to kind of expand on this fairly old technology with respect to adapting this to the needs of transposition. So all you have to do is now you clone your gene of interest into a Sleeping Beauty vector. Again, you maintain this gene of interest in the form of a plasmid DNA, and then you mix in a little bit of transposase encoding messenger RNA that you prepare in vitro, in vitro transcription reactions. So you mix this RNA and DNA together, and then you do this typical pronuclear microinjection step, and then you derive transgenic animals, transgenic mouse in this particular instance, that grow out, at least in a number of cases, out of these microinjected embryos, so that they give rise to animals where the gene of interest, in this particular case, a venous fluorescence protein, would be expressed all over the body of this animal that develops out of the microinjected embryo. And the efficiency of this step is really so high, about 50%, which means that every second mouse that will be born out of such microinjected zygote will really be a transgenic animal expressing this protein all over its body. Every second animal will be transgenic, not only in somatic tissues, but also in the germline, so that this animal can really pass on this genetic information event in the germline through the next generation of animals. And I'm also making another point beyond efficiency, and that really is expression of transgene in transgenic animals. So first of all, you see here transgenic pigs that were engineered by Sleeping Beauty Transposition, and again, a microinjection technology. And what I'd like to draw your attention here is really that these animals that contain 2 copies of the Sleeping Beauty Transposon develop a stronger phenotypic appearance, a more intense fluorescent signal than another transgenic animal that has only a single integration event in the genome. And the same thing up here in the upper panel, what you see are mice, transgenic mice, that contain varying numbers of integrated Sleeping Beauty Transposon copies, 1, 2, 3, 4, and again 3. And again, the point being is that those animals that contain higher copy numbers apparently express more intensively their genetic cargo. So I'm referring a little bit back to the cell culture experiment that already kind of indicated to us that maybe not all, but a very significant number, a very significant fraction of these integrated transposons express their genetic cargo long-term in the targeted cells. Now, as I already mentioned, I'd really like to tap into therapeutic cell engineering, which is kind of a very dramatically and very dynamically evolving and developing area of using transposons for genetic engineering. The first slide that I really want to show to you is kind of a summary slide that shows to you how we apply Sleeping Beauty Transposon-based genetic engineering in humans. And that is what we call ex vivo gene therapy. Now, what does it mean, ex vivo engineering? Ex vivo really means that we, first of all, isolate cells from a patient. Yeah? So ex vivo means that genetic engineering of patient cells doesn't occur in the body of the patient, but outside of the body in a petri dish or in a flask in a laboratory. So after isolating patient-derived cells, we would now take Sleeping Beauty vector components equipped with therapeutic genes. And we typically would use an electroporation technology to incorporate this novel genetic information in the isolated cells. Then these cells would undergo some enrichment and some cell culture for about a week or two, and isolating the cells and enriching this cell culture in the genetically engineered cells. Typically, of course, in terms of gene therapy, every human application would be preceded by a full validation of this experimental procedure in preclinical animal experiments for efficacy and safety. But of course, down the line, the entire circle is closed where these genetically engineered cells are reinfused back into the patient in the hope of observing some therapeutic benefit. That's kind of a summary how ex vivo gene therapy is made by Sleeping Beauty Transposons. And I'd really like to give you now 3 examples of how this system can be used for gene therapy, and 3 different cell types really that we want to target with these gene therapy experiments for the goal of fixing and providing therapeutic benefit for different kinds of diseases, really. And the first example really leads me to a disease of the eye that affects the vision, typically kind of a pathology that is affecting the life of the elderly population. It's a neurodegenerative disorder that we call age-related macular degeneration. So it's typically affecting the older generation, and it's cumulative in a sense that it starts with a certain impairment of vision. I highlighted here on this photograph, on this picture, what you would experience if you had AMD, and in the real serious cases, this can lead to a total blindness of patients. So let me just give you a few words with respect to the biology of this disease, AMD. AMD is really an outcome of a miscommunication or an imbalance of signaling mechanisms that regulate blood vessel formation of the retina. And these 2 signaling pathways, and the main components are VEGF and PEDF, that kind of maintain this balance of a healthy blood supply of the retina for the proper functioning of the photoreceptors for vision. And what happens during AMD is that this balance is completely broken, and there's kind of a very dominant role of VEGF that leads to kind of an overexpression or an excessive blood vessel formation at the retina that kind of damages the photoreceptors, leading to AMD and loss of vision. So the current medical intervention to kind of treat this is to provide an antibody against VEGF. And this really is a transient injection of the antibody directly into the eye. About 1/3 of the AMD patients are not responding to this antibody injection. That's number one problem. And number two problem is that obviously an antibody injection is by nature only a transient solution, so that these patients really have to show up at the clinic every single month to receive an injection into the eye. So this kind of told us that maybe we could put together a gene and cell therapy based approach for AMD that could provide long-term benefit for these patients. The idea here is the following. A gene and cell based therapy for AMD patients is based on the following concept. Cells from the patient eye, not from the retina, but from the iris, and these cells are called IPE cells, or iris pigment epithelial cells, can be isolated from the eye through a very small biopsy. This can be cultured for a short period of time in an appropriate vessels and tubes. Then an electroporation step would take place that would provide a therapeutic gene. And I already kind of highlighted to you the other component of this very fine balancing act at the retina, which is PEDF. PEDF is an anti-angiogenic factor. What this would do is that this would suppress blood vessel formation at the retina when overexpressed and distributed in the patient eye. So we take a Sleeping Beauty Transposon that has a PEDF transgene in it. We have shown that we can incorporate this transposon vector into IPE cells very efficiently. And these cells express human PEDF for long periods of time. And then therapeutically, the concept is that these cells, following ex vivo gene manipulation, would be really transplanted into the retina of the patient's eye, where this would sit down as a clump of cells and kind of permanently release PEDF in the local environment. The hope being that once PEDF is released in the local environment, this would suppress blood vessel formation and would prevent further damage to the photoreceptors. And this is shown here on this slide with an animal experiment, a rat model, where one can induce damage to the retina and then follow the fate of this damage in the context of neovascularization, basically a quantitative measurement of blood vessel formation directly at the retina in vivo in an animal model. And what we have seen in this animal model, that those animals, those experimental animals that received a transplant of genetically engineered cells expressing PEDF, do actually show a shrinkage or a reduction of this area where blood vessel formation can be measured. And this can also be quantified here. And this really is kind of the in vivo preclinical evidence that kind of suggests that this system can be useful later on in clinical trials in AMD patients. And we're all very excited about the future clinical development of this technology for AMD patients. So this was example number one. Let me now give you another example that really targets a completely different cell type, which is the hematopoietic stem cell. Obviously, those genetic pathologies and diseases, inherited genetic pathologies that affect the blood, for example, an immunodeficiency, can potentially be fixed by hematopoietic stem cell engineering. What this really means, experimentally speaking, is that these hematopoietic stem cells can be isolated from the bone marrow or following mobilization into the peripheral blood, these cells can be isolated and genetically manipulated so that a missing function or can be added, or a faulty gene can be repaired. And then this genetic modification can be passed on onto every single effector cell type that differentiates out of these hematopoietic stem cells following transplantation of the genetically manipulated cells back into the patient. So again, it's kind of a typical ex vivo application where genetic engineering takes place outside of the body of the patient. But in this particular case, we're targeting a stem cell type so that every single cell that differentiates out of these cells, either a T lymphocytes or a macrophage, anything practically speaking, would carry that genetic information that we introduced into the genome in the stem cells. And here's an experimental a slide that shows to you how we manipulated the Sleeping Beauty Transposon components so that they can be really useful for human hematopoietic stem cell engineering. Why did we have to kind of look for a modification of the system? The main argument behind this was that plasmid electroporation is very, very toxic in this particular cell type. So we argued that we could possibly derive smaller versions of the Sleeping Beauty vectors. For example, for the transposase enzyme, derive a variant of the plasmid, which is called a mini circle. A mini circle is also a circular molecule, just like a plasmid, but it doesn't have kind of the plasmid backbone any longer. So a mini circle is, practically speaking, nothing else but an expression cassette in a circular form. So we made these mini circles for a transposon that has a venous reported gene, as well as for the SB100X hyperactive transposase. And we also tested, just like in the animal transgenesis experiments, the performance of synthetic messenger RNA in electroporation reactions. And the very first kind of technically important observation that we made is that once we combine a minicircle transposon together with a transposase RNA, these two molecules, the toxicity of this electroporation reaction is lower than with plasmid constructs. So we actually do recover a fairly decent, fairly significant percentage of living cells after this kind of genetic modification system. Obviously, it's not only toxicity or the survival of the cells that is important in this particular case, but also how well and how significantly we can express a foreign gene. And again, what we see is that once we combine a minicircle transposon vector together with messenger RNA, this results in significant frequencies of those cells that express our fluorescent marker. And also, this is shown here on the right-hand side, about 50% of our cells do express this venous fluorescence protein for long periods of time in CD34 positive human hematopoietic stem cells. Importantly, these cells can be transplanted into experimental animals, immunodeficient mice that we use as recipients for engrafting human hematopoietic stem cells into them. Here on the left, what is shown is that human cell engraftment can be detected in these animals 3 months post-transplantation. That's number one. And number two, a significant fraction of these cells do express the venous marker following transplantation. So that means we can not only manipulate these cells in a relatively non-invasive manner, but these cells also maintain a stemness character so that we can transplant them into an animal. And these cells can not only survive there, but can also engraft and give rise to differentiated progeny in these experimental animals. So this was example number 2. And example number 3 really is CAR T-cell development. Now this leads us to T lymphocyte genetic engineering in the context of cancer therapy. How do these CARs really work? A CAR is an abbreviation for chimeric antigen receptor. These are really fusion proteins where an antibody domain recognizes a tumor-specific antigen. CD19 is a very well-known antigen in a B-cell-derived leukemia and lymphoma. So that's kind of recognition domain of the fusion protein, and the fusion partner is a signaling domain, so that if a T lymphocyte expresses this chimeric antigen receptor, it can recognize and bind to a surface antigen that is displayed by the cancer cells, and then at the same time undergo specific cell killing and eradication of the cancer cells as a kind of direct outcome of this primary recognition. Let me now give you kind of an outline about certain products that are also out there already targeting CD19 and some other antigens that are approved as products out there on the market. All of them use viral technologies to engineer T lymphocytes, lentiviral or retroviral gene vectors. However, it is projected that in the future, there will be a tremendous growth, almost kind of exponential growth of gene and cell therapies out there globally, which kind of forecasts that manufacturing efficient, safe, and economically competitive gene therapy products will be key to meet the increasing demands in the future. And we really do believe that transposon vectors kind of contribute in a very significant way to the development of some of these novel technologies. And I just want to kind of summarize on one slide what we did with Sleeping Beauty vectors in the context of CAR T-cell engineering. Again, we used these minicircle-derived vectors, electroporated these minicircles into human T lymphocytes, CD8 positive T lymphocytes. As you see, over 50–60% of these T lymphocytes do express the CD19 CAR. In this particular case, the gene of interest is CD19 CAR. And finally, in vivo validation shows to you that in an experimental model where you kind of detect CD19, the growth of CD19 positive human cancer cells, lymphoma cells, these cells can be fully eradicated by infusion of these genetically engineered CAR T cells. Basically, this cancer is gone, which really means that these Sleeping Beauty vectors are suitable for making biologically active cell products, and these provide certain advantages in CAR T-cell development. There are a couple of clinical trials already ongoing in CAR T-cell engineering, and I just wanted to highlight one multi-center clinical trial in Europe, which is called Caramba. This clinical trial is aiming to use Sleeping Beauty engineered T lymphocytes that target an antigen, a tumor antigen that is called SLAMF7 in the context of multiple myeloma. This clinical trial is already ongoing, and we are all excited about the prospects of these new developments. And finally, I'd really like to, in the past, or the last couple of minutes, I would just like to highlight one particular area that is fundamentally important whenever we talk about any kind of genetic engineering technology in human cells for therapeutic purposes. It's not only relevant for transposons, for every other technology, but transposons are not exceptional in that case. what do we mean by this? Transposons are natural mutagens. Transposons were discovered by Barbara McClintock, very famous experiments in the maize system, observing these kind of variegation in the color or the pigmentation of the maize kernels. And then this phenotypic variegation was really the outcome or the manifestation of a transposable element hopping in and out of certain genes. And this was really the biological basis of the discovery of mobile genetic elements in Barbara McClintock's experiments. So it's clear that transposons can also be potentially mutagenic in a human experiment, in a gene therapy application. What potential mutagenic activities could be there with a transposon integration? So first of all, a transposon could integrate into a coding sequence, for example, into an exon, and then would knock out the gene, kind of leading to a loss of function phenotype. Or a transposon could also integrate into a regulatory region, transcriptional regulatory region of an endogenous gene, and could kind of transcriptionally override the transcriptional program of such genes, leading to gain-of-function mutations of an overexpression phenotype, which is also unwanted. Let me now share one slide with you that kind of highlights how different integrating genetic elements are distributed across the genome. Namely, a retrovirus vector in yellow, a lentiviral vector, HIV-based vector in red, a piggyback transposon in blue, and Sleeping Beauty Transposon in green. And what you do see here is that transcriptional start sites, gene sequences, exons, introns, CPG sites tend to be overrepresented in the genomic integration sites. Lentiviral vectors have been very well known to target expressed genes in targeted cells, whereas retroviral vectors and piggyback transposon vectors tend to target the transcriptional start sites or the promoter regions of genes. Neither of these two sites in the genomes are compatible with safe integration. As you see, Sleeping Beauty is more or less randomly distributed across the genome, so there's more or less a random chance that the Sleeping Beauty transposon would hop inside a gene. And in the last couple of minutes, I would just like to share with you some very, very recent work that we did with the goal of actually trying to direct Sleeping Beauty transposon integration away from genes. In other words, to make Sleeping Beauty integration safer in the context of human applications. What served as a basis of this work is a structural information, a crystal structure of the Sleeping Beauty transposase in a model together with the target DNA. This is coming from Orshaya Barabash lab at the AMBL in Heidelberg, Germany, and some modeling work, structural modeling work that was used to highlight similarities of the Sleeping Beauty transposase and its interaction with the target DNA and another transposon, which is called MOS1, and retroviral integrases. And the direct outcome of this structural modeling work is that we highlighted 3 amino acid residues in Sleeping Beauty in position 187, in position 247, and 248 that are situated in a 3-dimensional structure very, very close to target DNA, that's number 1, and very, very close to amino acid residues in transposases and retroviral integrases that have been shown before to be involved in target site selection of these genetic elements. So the experiment that we did, and that's what I'm showing on the next slide, is that we subjected these 3 amino acid residues to mutagenesis. We basically replaced all 3 of these amino acids to everything else that is possible. And then we surveyed and scored the transposition efficiency of these mutant transposases for the 3 series of mutants. And as you see, for 187, these transposases are fairly efficiently supporting transposition. 247 and 248 are fairly sensitive to mutagenesis because only a few of these mutants have really displayed any integration efficiency. The next step was that we took a few of these, we kind of selected a few of these mutants and asked, where do integrations occur on a genome-wide scale when we use these mutant transposases as opposed to SB100X? And here's something really important discovery. When we look at Sleeping Beauty integration in the host cell genome, Sleeping Beauty really targets a consensus sequence that is composed of 80 dinucleotides. 80, 80, 80, 80, where the central T8 dinucleotide is the actual integration site. However, you have to kind of select out hundreds or even thousands of these integration sites and then put them into a kind of a big sequence alignment in order to see this particular sequence motif. Indeed, only 2% of all Sleeping Beauty transposition events occur in this particular sequence element. Now, some of the mutant transposases that we analyzed, namely this particular mutant, which is a proline residue sitting in 187, and then a valine, and then also an arginine sitting in 248, have this displaying an overrepresentation of integration into this particular sequence motif. Almost 40% of this mutant lands in this particular sequence element, in this A2 repeat. This is a gain of specificity with these mutants. Where do these integrations actually occur in the genome? Again, very briefly, just want to highlight two important findings. One up here, when we use these mutant elements, especially the 248 arginine mutant, this mutant directs integrations away from genes. What I highlighted here are coding exons. If you see, random frequency is designated a value 1, and then this particular mutation targets exons at a frequency of 0.2, which is basically a 20% a level of integrating genes into an exon of a gene. And down here is highlighted chromatin-defined functional segments. It's kind of the second observation that shows to you, again, I would like to highlight this particular mutant, again, a very significant depletion of integration into enhancer, as well as transcriptional start sites, transcriptional regulatory elements of genes, not only in relation to a retroviral vector, but also in comparison to a fully random distribution of sequences. And kind of the biochemical explanation for this is that apparently these mutant transposases prefer flexible DNA sequences where DNA structure maintains a bendable and flexible structure. So what is highlighted here is that as opposed to Sleeping Beauty transposase, these mutants really prefer a sequence environment that is associated with bendability. And of course, the 80-rich DNA or these 80-80-80 repeat sequences are typically associated with such bendability. And these three other panels really show denaturation energy, how much energy is required to actually pull apart the two strands of the DNA, and then stiffness or rigidity. And apparently, for all three measurements, our Sleeping Beauty transposase variants show that these integration events are associated with a highly flexible and bendable structure. That leads me to summarize my presentation. First of all, transposons are plasmid-based, non-viral delivery vectors for gene transfer, and I already kind of highlighted a few examples for you, and also highlighted, for example, the Karamba clinical trial, where in the context of cancer, cell therapy, Sleeping Beauty is already on the clinical testing. Sleeping Beauty transposon vectors combined with minicircle technology allow efficient, stable engineering of therapeutically relevant human cells, including not only T lymphocytes, but also hematopoietic stem cells. With respect to genomic integration, Sleeping Beauty has a close to random integration profile and thus might be safer than kind of usually or widely used retro and lentiviral vectors in human gene therapy. And in very recent work, we highlighted the significance of 3 amino acid positions in the Sleeping Beauty transposase and made some mutant variants of the transposase that kind of direct integration away from gene sequences, thereby enhancing the safety of Sleeping Beauty engineering in the context of gene therapy. And finally, GMP cell manufacturing of non-viral vectors has a competitive and economic advantage over viruses, and therefore believe that this kind of non-viral genetic engineering technology has a bright future for engineering therapeutic cells. Finally, that leads me to kind of highlight and acknowledge the input and hard work of my research laboratory at the Paul Ehrlich Institute in Langen, Germany. Also acknowledge the input of some collaborators, and would like to also thank you for your very kind attention. And if there's still some time for questions, I'm looking forward to them, and I will be happy to answer them. Thank you very much.
Thank you very much, sir, for this informative talk. We recently had in our coursework about Sleeping Beauty, and listening to its applications and how it works was very informative and very understandable. Now I invite any questions from the audience.
Could I start, please? Is that all right?
Yeah, sure.
Thank you, sir, for the wonderful talk. I had 2 questions for you. One was concerning the rate of remission of CD19 cancers after CAR therapy. So do you have any, could you comment about what the remission rates might be? And the second thing was, you mentioned that you could kind of play with the copy number to increase the expression of a particular gene in animal models. So I was wondering, how does that affect, how does the copy number affect, let's say, angiogenesis in your AMD model? So if you play with the copy number, can you actually reduce angiogenesis even further? So those were my questions.
Yes, very, very good questions. Thank you very much. Now, with respect to remission, you know, it's not really, or let me put it this way. Currently, there's no clear implication or evidence that the actual technology through which you manufacture CAR T cells would have such a profound effect on remission. My understanding is that remission is largely contributed by loss of antigen expression in the patient. So, for example, in the context of CD19 cancers, the target cell population, or at least a fraction of the target cell population, would undergo antigen loss. And then obviously, although you have CAR T cells in the circulation, those cells cannot be targeted. Those target cells that lost antigen expression cannot be targeted any longer. Probably antigen loss is something where you would argue that no matter what kind of technology you're actually using, either transposon or virus or a CRISPR-Cas engineered cell population, those CAR T cells will not be able to really target those cells any longer. And therefore, one really needs to kind of bring up some additional technologies to target these cell populations. So just like to mention 2 of them. For example, some epigenetic kind of a treatment of the cells so that antigen expression is not so profound in the targeted cell population. In other words, CD19 would keep expressing for longer periods of time in a larger cell population. And second, kind of targeting not only one tumor-associated antigen, but 2 simultaneously. So you could argue, although one is kind of losing expression, but there's still another antigen that is expressed on the cell surface, so that there's kind of a fallback option in vivo to also target those cells. So that's one thing. Now, in the context of the AMD experiment, that's very good, excellent question. Obviously, the more PDF you secrete in the eye, in the local environment, the higher therapeutic benefit you could kind of logically expect. The thing is, it's kind of a sensitive balance that you have to kind of keep. Vector copy number with respect to expressing as much therapeutic protein as possible, that's great. But what you have to also keep in mind is kind of the last segment of my presentation. The more copies you have in the genome, the higher the risk you're entering, because once you're kind of transplanting more and more copies across the genome, you're also multiplying the relative risk of actually hitting a gene where you could end up with some kind of an unwanted adverse event in that cell population. So it's kind of a sensitive balance with respect to a therapeutic benefit that you want to get out of that engineered cell population, but at the same time keeping vector copy numbers as low as possible so that you're not entering a very risky area with respect to doing something to the genome that should be avoided at all costs. So right now, in practical terms, in these engineered cell populations, we're really trying to keep vector copy numbers below 5 per cell, so that we're not really entering a very risky kind of window of vector copy number, but at the same time still being in a therapeutic window of where we can expect a therapeutic benefit for the patients.
Thank you. Fantastic talk. Thank you.
Does anyone else have any questions? If not, I think we would like to end the talk now. I would like to thank everyone who attended this talk and got actively involved in the interactive session. I extend my gratitude to our faculty advisor, Dr. Sadananda Singh, and the School of Biology for their continued support towards Proteus. Wishing everyone a wonderful day ahead. Thank you very much. Thank you, Professor.
Thank you.
Thank you.
Thank you. Bye.
Bye.